So, you're leaving already?
No, I'll be like right back.
So I wanted to back up just a little bit. We're talking about we've been going through the endocrine system and all the endocrine structures, tissues, glands, organs, the hormones they produce, their mechanism of action to a point. What's this? That was weird. Mechanism of action, what the hormones do from a systemic standpoint, and now we're talking about, we left off talking about last time the integrated stress response, what our body does from an endocrine standpoint to adapt to stress. And again, we're for the most part talking about stress being, just something happening in our body where we're out of that homeostatic window. Our temperature has gone up above 100 degrees Fahrenheit or 38 degrees centigrade. pH is increased or decreased above that very narrow pH range for most body systems. Blood pressure, heart rate going up or down. That's what I'm talking about, a stressor situation and what our body does to adapt to that or to compensate for that. Okay, and it's a multifaceted response, bringing into play many different hormones, many different systems, and some non-hormonal systems. But one of the primary reactions to stress is right here, the initiation of the fight or flight response. Sympathetic innervation, sympathetic activation, okay, directly from the hypothalamus, signaling to the adrenal medulla to release epinephrine and norepinephrine to play a role in that fight or flight response, to get our body ready to go into action and to inhibit those systems that we don't particularly need at that point, like the digestive system, reproductive system, urinary system. Okay, peripheral blood flow. So that's the big one, the sympathetic nervous system activation. And whenever you talk about, the sympathetic activation, you're also going to be talking about this cortisol system. Cortisol from the adrenal cortex and its play, and also when you're talking about the sympathetic nervous system, sympathetic activation, remember the thyroid system is also going to be playing a role in that, because the thyroid hormone plays a role in the in a nutshell, the increased metabolic activity of cells. Okay. So there's many different hormonal systems that come into play, and again, the cortisol system and the sympathetic nervous system, it all starts from the level of the brain, the hypothalamus, because like I said, the hypothalamus is hardwired into the adrenal medulla, and this is also what upregulates the cortisol system through the release of corticotropin-releasing hormone from the hypothalamus that stimulates the anterior pituitary to release adrenocorticotropic hormone. That stimulates the adrenal cortex to do a lot of things, but in terms of cortisol, the release of cortisol from the adrenal cortex. Okay? Oh, I forgot to get this. And so the specific stress on the body is going to initiate two things: a specific response at the local area, okay, and then a system-wide, just general response to this adaptation syndrome, if you will. Okay, and so this very busy slide shows us what happens when we are under stress, irrespective of what kind of stress or the degree of that stress. And what we have here is it starts up top with whatever the stressor may be, And again, that action is directly communicated through the hypothalamus. And in terms of the cortisol system, it's going to, the hypothalamus itself is going to release corticotropin-releasing hormone, whose target tissue are corticotropes on the anterior pituitary. That hormone, CRH, moves down into the environment of the anterior pituitary through the hypothalamo-pituitary portal system, stimulates corticotropes to release ACTH, adrenocorticotropic hormone, that stimulates the adrenal glands, specifically the adrenal cortex, to release cortisol. And then what cortisol does is, again, part of that fight or flight response, not part of the fight or flight response, but goes hand in hand with it. And what's happening here is it's going to mobilize energy stores, bless you, specifically, for example, like the glycogenolysis, the breakdown of glycogen to release free glucose for cells to utilize for cell respiration and the yield of ATP. Okay. And also, it's going to initiate the sympathetic activation. Again, the hypothalamus is hardwired into the pancreas, not hardwired, but connected to the pancreas as well through the sympathetic innervation. And what the epinephrine and norepinephrine are going to do at the level of the pancreas, the endocrine pancreas, is to stimulate the release of glucagon and inhibit, in part, insulin. And that might not make sense initially, because if we're going to need glucose for increased metabolic activity, and insulin sensitivity is, what's happening on the thing? Insulin sensitivity of cells is required for mobilization of glucose into the cell. That might seem counterintuitive. But what's happening here is the glucagon is being released to free up energy stores, particularly the breakdown of glycogen and release of free glucose. So at this particular time, insulin is going to be inhibited. And we'll find those two hormones are always in flux. One is high when the other one's low, and vice versa. So it's not a direct inhibition, it's just a depression of the insulin activity for a moment. Okay. Again, and now in the adrenal medulla, we're going to be getting the release of that epinephrine at the level of the vasculature. It's going to result in vasoconstriction. The vasoconstriction, the point is to increase blood pressure. But one effect of that the effect of that vasoconstriction is to reduce blood flow into through the kidneys. Okay, and again, that sounds like a negative thing, but that decrease in blood flow through the kidneys. Now, remember, we're not really worried about the urinary system, so decreasing blood flow to the kidneys is not a big deal. But what this does as a positive effect is that decrease in blood flow is sensed through the afferent arteriole, and that initiates the renin-angiotensin-aldosterone system. Okay, and that's going to release aldosterone, and aldosterone is going to play a role in increase sodium reabsorption, but the point of that is to also increase water reabsorption, to increase fluid balance or fluid volume, increasing fluid pressure, and thus increasing blood pressure. Okay, and also from the hypothalamus's interconnection to the posterior pituitary, we're going to get the release of ADH, vasopressin, which is going to increase water reabsorption in the kidney, specifically at the level of the distal convoluted tubule and collecting duct, where we're going to insert more aquaporins, increasing dramatically water reabsorption, and again, another level of increased fluid volume, increased fluid pressure, increased blood pressure. Okay, and so all this is going on in that response to stress, irrespective of what the stress is or the degree of that stress. Okay. And so we can see that it's a multifaceted response. It's involving multiple hormones at multiple levels in our body, all over the body, okay? Again, it doesn't matter what the stress is. The problem with, or the potential problem, I should say, with this reaction to stress is down here. Stressors that aren't something that's out of that homeostatic, has nothing to do with trying to maintain homeostasis in the body. Psychosocial stress, getting stressed out before exams, you know, breaking up with your significant other, you know, getting caught in traffic. All those stressors are stress, and our body's going to react the same way. But it's going to react the same way when our body doesn't really need it. Like if we are under stress because we've got a fluctuation in blood pressure, let's say our blood pressure drops and our body goes into this stress response. One thing that's going to happen that we just saw is a lot of water, additional water reabsorption, right? The vasopressin from the posterior pituitary, aldosterone from the adrenal cortex, peripheral vasoconstriction, you know, all these mechanisms to increase blood pressure, the fight or flight response, okay, increase metabolic activity. There's a benefit for that. But if we're just having, you know, a really shitty day, you know, and we're just got exams and projects are doing all that kind of stuff, our body's reacting the same way. We're going to be reabsorbing a lot of water, you know, or we're going to have an increased heart rate. We're going to have increased blood pressure. And that can have a negative effect because we didn't really need that in the first place. And so what happens is we can potentially be having all these systems initiated and activated and our body doing certain things because of this stress response that we really didn't need to have happen anyway. Now, in the case of, you know, breaking up with your spouse or getting caught in traffic or, you know, those, or even, you know, getting stressed out over exams, those are, you know, periodic things. And so our stress response isn't really going to, even though we don't need all those different things to happen, it's not necessarily a big deal because it's a, you know, one-time only sort of situation. But if you're under chronic stress for over periods of time, it can take a serious toll on your body, okay? Let's say you are, what's the phobia for just being afraid to be out in the open? Does anybody know? Is that agoraphobia? You know, say you have that, and you have to be out because you have to go to the store, you have to work, you got to, you know, be out in the open. So you're constantly under this just incredible stress, and your body's reacting the same way as it were if it were a blood pressure problem, okay? That takes a serious toll on the body over time, okay? And also to point out, right here's a big one. Chronic stress, and specifically the release of cortisol, has an effect on the immune system. It has an anti-inflammatory effect, but even more important, has an immunosuppressive effect. It suppresses our immune system. So again, over time, this can have very negative outcomes, okay? So like I always tell my students, I think I've already told you all this before, whenever you're stressed out, just quit, stop, take a deep breath. Let it go. It's that easy, right? Y'all are all looking at me like, whatever. It's a joke. I know you can't just let it go. Okay, so moving on down. The pancreas, specifically the endocrine pancreas. We talked about the exocrine pancreas in digestion, and now we're going to talk about the endocrine pancreas. And there's only a small portion of the pancreas that is endocrine, like about 1% of the total mass is actually tissue that plays an endocrine role, but a very important endocrine role. When we're talking about endocrine pancreas, we're talking about regulation of metabolism, okay? And metabolism can be broken down into anabolism, catabolism, and then conversion of molecules into other molecules, if you will. And I'll give an example of that in here in just a second. But you know, anabolism is building up. You know, taking at the molecular level, taking organic molecules and putting them together and building something up. Like that glycogen formation we talked a minute ago, taking free glucose, packing it up into glycogen molecules, that's anabolism. And then catabolism would be an example, breaking that glycogen down into free glucose. I always tell my students the way I remembered anabolism. I know you guys don't really have this problem remembering what's what, but I always told them that anabolism is building up, and always think of anabolic steroids. You know, bodybuilders take anabolic steroids to build up muscle mass. And I always tell them that they probably look at me and think that I'm on anabolic steroids because of, you know, this. But this is all due to hard work. Okay? I'll get y'all to laugh sooner or later. Interconversion, just to clarify, what we're talking about in addition to anabolism and catabolism is small molecules not coming together to form like a larger, like glucose, individual glucose molecules coming together to form glycogen, which is anabolism. This is small molecules being converted into other things. It's still part of the metabolic mechanism or pathways. And an example is amino acids can be transformed into glucose and fatty acids. Specifically, and we'll talk about this in more detail as part of the endocrine pancreas, but one effect is something called gluconeogenesis, production of new glucose. When our body is in need of a potential energy source and free glucose and other carbohydrates and fatty acids are scarce, one thing it will turn to is gluconeogenesis, where it can take some amino acids and not many free amino acids and form glucose. Okay, not much, but it can. And what the body does to obtain those free amino acids is start breaking down protein. And so it's a process called protein wasting. And so when our body has turned to that as its sole source of glucose, you're basically in a starvation situation. But anyway, that's an example of interconversion, converting molecules into other molecules. Okay, let me, this should have, well, it's not working on this. This was a video, an animation, and I don't even know if it was a good one. I put it in yesterday. I haven't looked at it, so, but it won't play on this for some reason, so we'll just keep on moving. So again, in terms of potential fuels, this is where they're coming from. Of course, we have free glucose. Glycogenesis is the genesis of that glycogen, where we're taking free glucose molecules and forming glycogen, and then that glycogen is stored in hepatic tissue and skeletal muscle. When we have free glucose in our system, our cells are going to use the available glucose, and there's always some that's left over, and our body will package that up and store it for later, and it stores it specifically in liver tissue, hepatic tissue, and skeletal muscle. Okay? And then when we're in between meals, glycogenolysis, we're going to break down that glycogen. Okay? Gluconeogenesis, like we said, take free amino acids and turn that into glucose. Protein synthesis, we can take free amino acids and obviously incorporate that into larger proteins, and then we can break those down as needed to free those amino acids and utilize those free amino acids in the interconversion into glucose and to fatty acids, because fatty acids are an excellent source of potential energy as well. They're long hydrocarbon chains, and anytime you have a molecule with a high number of hydrogens relative to everything else, there's a lot of potential energy in there. And so the fatty acids are excellent sources. And then, so of course, fat synthesis, as a storage form of those fatty acids, and then breaking them down to release them as a energy source. And again, fatty acids are an excellent, excellent energy source, and actually yield a little bit more ATP than glucose does. Okay? We need a way to store this potential energy, because where does this energy originally come from? Well, it comes from the stuff we put in our body, through that process of digestion, breaking up that food into its individual nutrient components that our body can utilize. Okay, we don't eat all the time, although sometimes we might think we do, and so we need a way to store that, those potential energy sources. Okay, and ways in which we have already highlighted. Okay, the glycogen, the lipogenesis, the formation of fats to form triglycerides, the amino acids, storing those as proteins and other molecules. And they all can be utilized as a potential energy source. Most cells in our body, they want glucose as their initial potential energy source, because glucose is one of those molecules that has a bunch of hydrogens relative to everything else, C6H12O6, twice as many hydrogens as carbons and oxygens. So there's a lot of potential energy holding that molecule together, and when we break it down, we start to release that energy that the cells can utilize. Okay? That's what cells look for first, because it's a ready-made, power-packed energy source, potential energy source. But if glucose is in short supply, most cells in our body can turn to other sources, and the first thing they'll turn to is other carbohydrates. And when carbohydrates are in short supply, then it'll start turning to the fats and breaking down those fat molecules, releasing those free fatty acids, and those fatty acids can be utilized as potential energy sources. And if we've gone through all of that, then we can start that protein wasting and breaking down proteins and freeing up available amino acids that can be utilized for gluconeogenesis. Okay? Most cells can do that. Some cells depend on glucose, and they can only use glucose. And some very important cells are the cells of our central nervous system. Okay, this says the brain, but the central nervous system, brain and spinal cord, rely on glucose and only glucose for cell respiration. Okay, and so when we have a situation like the fight or flight response, or a response to stress, or sometimes in just everyday metabolic activity, that our day-to-day stuff, our body will switch into what's called a glucose-sparing effect. And what that means is we have a way to tell different tissues in our body, say those tissues that can utilize other sources, hey, you guys leave this glucose alone, you go after whatever else you can find, let's save that glucose for the central nervous system and other tissues that rely on glucose, and that's called glucose sparing. Okay. So these potential energy sources are stored and released in what are called absorptive states or post-absorptive states. Okay. The absorptive state is referring to the state that's just after a meal, and we're absorbing nutrients. Okay. And then the post-absorptive state is when we're in between meals, and we are, say, going into those energy stores, like that glycogen, like that breaking down fat molecules into free fatty acids, things of that nature. Okay. And there's some important tissues, and even though we're talking about the pancreas, there are other tissues that are involved in these absorptive or post-absorptive states. The liver, fat tissue, muscle, specifically skeletal muscle we're talking about, and the central nervous system. Okay. The liver is an important tissue because this is the primary tissue where we store that glucose in the form of glycogen. And like I said, it's not just liver. We also store glycogen in skeletal muscle as well. But the liver is the primary tissue where we store free glucose in the form of glycogen. And then obviously the main tissue where we release free glucose from the process of glycogenolysis. Okay. Fat tissue is a primary energy storage site because it's fat. Okay. This is where those fatty acids come from. A fat molecule is composed of a 3-carbon glycerol with fatty acids attached to one or more of those glycerol molecules through a covalent interaction. Okay, and so it's a primary energy storage site. Muscle is also a storage site for glycogen, like I said, but more importantly, we're talking about protein. That's what muscle is, and so this is an amino acid storage site for potential energy sources. And then the brain is important because, again, this is one of the tissues that only can utilize glucose. And more importantly, to make sure that we have available glucose for the central nervous system is that there is no glycogen stores in the central nervous system. There's no way to break down glycogen within the central nervous system and make free glucose available. Okay, and so that's very important to ensure that there is plenty of glucose on hand for the continual metabolic activity of our central nervous system. We can also tap into what are referred to as lesser energy sources, and they're referred to as lesser energy sources because the energy yield isn't that great as you would get with free glucose or fatty acids or gluconeogenesis or whatever. Okay, and those lesser energy sources are glycerol, lactic acid, and ketone bodies. Glycerol, like I said, is a 3-carbon compound, and it is the backbone of fat molecules, and it's the unifying feature of all fat molecules. And it's the same 3-carbon glycerol in every single kind of fat molecule there is. It's those long hydrocarbon fatty acid chains that define what specific type of fat you're talking about. Those long hydrocarbon chains are usually on the order of 16 to 18 carbons in length. They can be single bond, double bond, so saturated and unsaturated fats. Those are what define what type of fat you're talking about. That glycerol is uniform in that it's the same molecule from fat to fat to fat to fat to fat. And that glycerol can be a lesser energy source because in the process of fat catabolism and the breaking up of those, the releasing of those free fatty acids to where you have just free glycerol, that free glycerol can be converted to glucose in the liver. Okay, so it's one of those interconversion things. Okay, and glycerol can also, I'm pretty sure that glycerol can enter into some level of the glycolytic pathway, that first pathway in cell respiration. I believe right before, I think it's glucose 6-phosphate breaks into 2 molecules of whatever's downstream. I think it can fit in right there and yield some energy as well. Lactate or lactic acid is a byproduct of anaerobic cell respiration. So in those tissues that can respirate anaerobically, lactic acid is produced, which has a negative effect on the muscle tissue itself, but that lactic acid can also be converted to glucose in the liver, hepatic tissue. So it's one of those interconversion molecules as well. And then lastly, ketone bodies. Ketone bodies is a collective term, and specifically it refers to, for the most part, acetone, acetoacetic acid, and beta hydroxybutyrate. predominantly. There's a few others, but those are the three big ones that are collectively called ketone bodies. And these are produced during that glucose-sparing effect when tissues that can use alternative sources for glucose do. And what happens is the process of utilizing these free fatty acids. These free fatty acids are oxidized to release energy, and through that those oxidation steps, it gets down to the level of acetyl-CoA. And if you remember, acetyl-CoA is a metabolic breakdown product of pyruvate, the last molecule in the glycolytic pathway. And that acetyl-CoA gets converted to ketone bodies. Okay, and the ketone bodies can have a negative effect because they can affect the pH of the system. It can drop the pH, make it more acidic. But these ketone bodies specifically can be utilized as lesser energy sources that can be utilized, further oxidized in cells that utilize the Krebs cycle. Okay, so they can be utilized for further cell respiration. And many of the cells in our body do utilize this pathway and this further conversion of acetyl-CoA into metabolic breakdown products where we further the energy yield. Okay, but they're collectively referred to as lesser energy sources. Okay, in terms of the endocrine pancreas and its effect on that mobilization of energy sources or storage of energy sources is dependent on the primary hormones coming from the endocrine pancreas, and that is insulin and glucagon, somatostatin, another one called amylin that we'll talk about in a minute. But these all come from structures of the pancreas that are the endocrine pancreas that are referred to as the islets of Langerhans. Okay, and in the islets of Langerhans you have a whole host of different cells, and each one produces different hormones like alpha cells, beta cells, delta cells. Okay, and we're going to get into insulin and glucagon here in a minute. What somatostatin does is, this says it inhibits digestion, and that kind of oversimplifies it. Somatostatin kind of regulates the insulin, glucagon levels to keep them in balance. Okay, it does inhibit the digestive system to a point, decreases the process of absorption in the small intestine, decreases gastric emptying from the stomach into the small intestine, but its major role is in the regulation of insulin and glucagon levels. Okay, and just down here, this is a picture from your textbook. Obviously, we're talking about the pancreas, which sits down here right under the stomach, and the endocrine pancreas are these small structures that are islets. Okay, and this is what it looks like from a histologic standpoint. And this is pretty easy to see. You know, this region right here that's bound, it doesn't quite take up the dye that's staining these tissues as well. But really, under the microscope, they're kind of hard to see. You need to have a trained eye. You need to know what you're looking for because they're very small. But nonetheless, that's what they look like. And within these islet cells, we have the different, or I should say within these islets, we have the different cells that produce the different endocrine hormones from the pancreas. Okay, what insulin and glucagon does is increase or decrease blood glucose. Our blood glucose should be about, fasting blood glucose, 100 milligrams per deciliter. We get above that, insulin's released. Okay, and the insulin's role is to get that glucose into cells, or just to decrease the blood glucose level, get it back down to the normal range. And if we drop below that basic 100 milligrams per deciliter, then glucagon is released to bring those glucose levels up. Okay, so they're continually in flux to maintain that blood glucose concentration. Okay, and what it does is it has effects over and above just its role with glucose. Okay, it plays a role in regulation of carbohydrates as a whole. It plays a role in fat molecules, and specifically, it plays a role in taking those free fatty acids and incorporating those into the glycerol molecule to form fat, so to decrease the free fatty acid concentration. Actions on proteins, it decreases the free amino acid concentration by initiating protein synthesis, and there's some other effects on amino acids we'll talk about here in a minute. It's not just protein synthesis. But again, what insulin basically does, insulin is released from the beta islet cells, and its basic role is to bring down that blood glucose. We're going to talk about those mechanisms of actions here in a second. I talked about some different hormones that we may or may not have heard of, and this is one of them, amylin, also coming from beta cells. And what amylin does is it kind of goes hand in hand with somatostatin. And just to let you know, somatostatin is also a type of protein we've already learned about. Remember we talked about regulatory hormones from the hypothalamus, inhibiting and releasing hormones, and we talked about growth hormone has growth hormone releasing hormone and growth hormone inhibiting hormone from the hypothalamus. That regulates growth hormone production and secretion. Well, somatostatin is growth hormone inhibiting hormone. Okay, and amylin kind of goes hand in hand with somatostatin. What amylin does is promote satiety, so we get that feeling of being full. It's inhibiting the digestive system is what it's doing. It slows gastric emptying, so it slows the movement, the muscular activity of the stomach, so it slows movement of that chyme from the gastric environment into the small intestine, because once it's down there, stuff's going to start getting absorbed. So it wants to slow that down. And suppresses glucagon. Okay? So we want to suppress the action of that hormone that's going to increase free glucose. Okay? So it wants to suppress the digestive system. Okay. And so what insulin does to lower blood glucose is to get that free glucose into cells so the cells that need it can utilize it as a potential energy source through the process of cell respiration to yield ATP for the cell and thus for the tissue and thus for the organism. Okay. It's going to also take the free glucose that's left over, if you will, after the cells are utilizing. The cells have all the glucose they need. All the cells are happy, but we still have excess glucose in the circulatory system. Well, insulin's also going to start the process of production of glycogen, packaging up that free glucose, covalently bonding them in a, I think it's an alpha 1,4 or a beta 1,4 linkage, and putting that glycogen into hepatic tissue and skeletal muscle. It inhibits the breakdown of that glycogen, because what would be the point? Why would you be storing it and breaking it down at the same time? Bless you. And it inhibits gluconeogenesis, the formation of new glucose from free fat amino acids. Okay, and also in the inhibition of that gluconeogenesis, it decreases the amount of free amino acids available for this process, and it inhibits the enzymes that are necessary for gluconeogenesis. Okay, so the net effect of insulin is to bring the glucose concentration, blood glucose concentration down, and to bring the concentration down of those other molecules that can be utilized as potential energy sources. Okay. So, factors that affect blood glucose, factors that increase blood glucose, and factors that decrease blood glucose are here, and these are generalized factors. Factors increasing blood glucose is the obvious, putting food in our body, of which glucose is a nutrient component. Okay, so we digest our food and absorb the nutrients. We absorb that glucose. That's going to increase blood glucose. Okay, and when we're in between meals, we're not putting it in, but our body needs glucose. Well, that process of glycogenolysis, breaking down glycogen, releasing that free glucose to be available for cells. Gluconeogenesis, conversion of those amino acids into new glucose. All of that is going to increase blood glucose. Factors that decrease it are the utilization of that glucose. Once we have it in our blood, now we're going to need to put it in our cells through the process of insulin secretion and its activation at the cellular level, getting that glucose into cells, so it's going to decrease blood glucose. Okay, we're going to take free glucose and form the glycogen, so just the opposite of what we had over here. And we can also have urinary excretion of glucose. And I put a red arrow in here because this is when things aren't going well. And when we talked a little bit about this when we talked about the kidney, we don't want to see any glucose in our urine, right, in a normal situation. All of the glucose that's dumped in the nephron, 99.9999% of it is reabsorbed at the proximal convoluted tubule right when it gets in the nephron. But this is one way to excrete glucose when we are hyperglycemic and our kidneys can't handle anymore. We've hit that renal threshold we talked about. Now we're going to have glucose appearing in the urine. Even though that's a negative outcome, that's still a way to decrease blood glucose. Okay. What's the stimulus for insulin secretion? What makes those beta cells release insulin? Well, it's the glucose concentration. Glucose concentration goes up, insulin is stimulated for release. I'm going to show you the pathway, specifically what's happening at the cellular level that initiates that, but that's the signal for insulin secretion. And the cessation of insulin secretion when blood glucose concentrations go down. And that's what it's referring to on this slide when it says negative feedback. When glucose concentrations drop below the appropriate level, that inhibits the process of insulin secretion from the beta cells. Okay? And it stimulates at the cellular level, at the level of that beta islet cell. It stimulates it for secretion through what's called excitation-secretion coupling. And it's very much akin to when we studied skeletal muscles, when we studied excitation-contraction coupling, it's an analogous situation. Okay. So I put this in yesterday, and what this is showing you is what's happening specifically at a beta cell. That's supposed to be beta. And what stimulates the release of insulin. Okay, what stimulates the cell to start producing insulin and release it. Okay, what happens is we need glucose to start this whole process, even though insulin is required for glucose entry into cells, most cells. We need glucose to start this whole process so we can start the process of glucose mobilization. And what happens at the beta islet cell level is glucose enters into the cell through a glucose transport protein, and specifically this is referred to as GLUT2, glucose transport protein type 2. Okay, we're going to talk in a minute about the predominant glucose transport protein in most cells in our body, and that's GLUT4. I think there's 14 different, so far, glucose transport proteins that have been identified. And and they're located on different types of tissues. For instance, GLUT1 is on the blood-brain barrier, so that glucose can get into the central nervous system. GLUT2 obviously is found here on beta islet cells. It's also found in the renal tubules. When we have some of those glucose co-transport proteins, the actual glucose transport protein is a GLUT2. GLUT3 is on neurons, and GLUT4 is, like I said, on most cells in our body that utilize glucose in the other ways. And it's actually the cells that have the GLUT4 are the only cells that are that require insulin to get the glucose into cells. You can see here on these insulin secreting cells, insulin's not required for the activation of this GLUT2. But what happens is when glucose is in the environment, it will bind to and utilize this transport protein to get inside the beta cell. And once it's inside the beta cell, it starts getting converted into forms that can be utilized in cell respiratory cycles. The first thing that happens is it's phosphorylated to give us glucose 6-phosphate. And then through oxidation steps, it yields ATP. Okay. Now, I don't know if that's just the glycolytic cycle or if it's the entire cell respiratory cycle. It doesn't matter. Through the oxidation of glucose 6-phosphate, we're going to yield ATP. And that ATP is harnessed by a ATP-sensitive potassium channel. And what happens when ATP binds to that channel is it closes the channel. It doesn't open it, it closes it. So no more potassium can leave the cell. So when you have an increase in potassium concentration inside the cell, because it can't get out now, because with that ATP enclosing the channel, it causes a change in the membrane potential, and what it causes is membrane depolarization. Okay? And through the change in this membrane potential, this depolarization event, it reaches a potential that activates voltage-gated calcium channels. And calcium channels open, and calcium pours into the cell, and then calcium does in the beta cell kind of what it does in the axon terminals. It causes the fusion of vesicles, insulin-containing vesicles, with the plasma membrane and the release of free insulin, so that insulin can now go to those cells that are insulin sensitive, which is many of the cells in our body, and the eventual activation of the glucose type 4 transport protein, GLUT4. Okay. And so I put this slide in yesterday, and this shows you a little bit more specifically of the signaling mechanism to get that GLUT4. When I say activate, what it specifically does is cause the upregulation of these transport proteins into the cell membrane, so they're available for the transport of glucose into the cell. Usually they're in a cell that's not utilizing glucose at that particular time. These transport proteins are internalized in these membrane vesicles. Okay, GSV. This stands for GLUT4 something vesicle. I forget what the S stands for. But to get this up into the membrane, what we have to have in terms of GLUT4 is we have to have insulin. So the insulin is just released through that mechanism we just saw from the beta cells. It enters into the circulation as all endocrine hormones do, and now it's going to bind to its receptor. And its receptor is a receptor type called a receptor tyrosine kinase, TRK. And what it is a receptor that's a transmembrane receptor. Without insulin, these individual entities are separated. When insulin binds, it causes them to dimerize, come together. And on the intracellular side of the receptor. Remember, this is a protein, so it's made of amino acids. And on the intracellular side, there are a cluster of tyrosine residues. And what happens when the insulin binds to the receptor and it causes dimerization, it autophosphorylates itself. It also has a kinase domain. And remember, kinase is an enzyme that phosphorylates other molecules. So it phosphorylates itself specifically on the tyrosine residues. It initiates a very complex signaling pathway. And even this pathway, even though it looks very complex, has been oversimplified. And the activation of all these docking proteins and eventually causes the upregulation of the glucose transport protein, the GLUT4 containing vesicle, causes it to fuse with the plasma membrane, expressing the glucose transport proteins on the cell surface so they can internalize that glucose, get it into the cell so that cell respiration can occur through utilizing this glucose. And over here on the right side, this is just another pathway utilized by the insulin receptor glucose transport protein. So there's multiple pathways, but it all starts with insulin binding, activation of the insulin receptor, and eventual upregulation of this vesicle into the plasma membrane to make the glucose transport proteins available for glucose transport. Okay, so what insulin does is lower blood glucose. So insulin is referred to as a hypoglycemic hormone. That makes it sound like it causes hypoglycemia. It doesn't. It just lowers blood glucose to its normal level. Okay, but excess insulin can have a negative effect, and a drastic negative effect, causing hypoglycemia. And if you have too much insulin, just think about what's going to happen. What does insulin do? It drops blood glucose. If you have too much insulin, it's going to drop it dramatically. And remember, we've got those tissues that require blood glucose. That's why we have to keep that fasting blood glucose at 100 milligrams per deciliter. We have to have glucose on hand at all times. And if we get a dramatic decrease in that blood glucose, those tissues that require it are going to suffer. And the first ones to suffer is the central nervous system. Okay? And this is no shit, guys. This can cause some serious problems. And you know what I'm going to do? I'm going to go off mic because I have...
What was your number?
I have no idea. I was... yeah. I don't, like I said, everything from sitting on the couch after that's foggy. Oh, and also remember her, when she took, well, afterwards. So that's what insulin does. Brings down glucose levels. And glucagon does just the opposite, really just in opposite ways. Everything that glucose does, glucagon does the opposite. Okay, so it's going to mobilize carbohydrates, increase the carbohydrate concentration in the blood, increase that glucose concentration in the blood, break down fats, release those free fatty acids. Okay, break down proteins to a point. Okay, we're not talking about protein wasting, but just making free amino acids available, the ones that can be utilized for gluconeogenesis. Okay, so glucagon is active, or released, I should say, during that post-absorptive state. Okay, when we're, again, in between meals. Okay, and so insulin and glucagon, it's not a case of one or the other. Insulin working all by itself, and then another time glucagon working all by itself. They're in continual flux. Okay. Going up and down, opposite of each other, regulated by that somatostatin. that growth hormone inhibiting hormone, also regulated to a point by that amylin and some others. Bless you. But again, what stimulates the release is the glucose concentration. Glucose goes up, insulin's released, and glucagon's inhibited. Glucose goes down, glucagon is released, insulin's inhibited, or goes down. Okay? We can also have too much glucagon too. Glucagon excess can do the opposite of what insulin excess can do. This will cause hyperglycemia. Okay? And this can be very problematic in diabetics, particularly type 1 diabetics, insulin-sensitive diabetes mellitus. Okay? Because if the glucagon and the insulin are both in flux with each other, okay, and when insulin goes up, glucagon goes down, and when glucagon goes up, insulin goes down. When we don't have any insulin, because of that type 1 diabetes, that autoimmune disease, we can potentially have increased levels of glucagon at all times. And what that will result in is hyperglycemia. And one of the first things that's going to be affected with hyperglycemia is neurological problems as well. You get too little glucose or too much glucose, and it's going to affect the nervous system first. Okay. In addition to insulin and glucagon maintaining those carbohydrate levels, or those levels of potential energy sources, I should say, we also have other systems as well. The sympathetic nervous system, fight or flight. You know, they're mobilizing glucose, right, in different ways. The cortisol, like we talked about. But it's all under regulation at the beginning of the day by the hypothalamus. Okay. Now this is a busy table from your book, but it's a really good one that kind of summarizes everything, so I kind of like it. But it lists all these important hormones that play a role in the mobilization of these potential energy sources and the different effects they have on the glucose itself, on the fatty acids, on the amino acids, on the proteins, all those potential energy sources. Okay, and if you understand what the hormones do or their cellular effect, then you can surmise what they do in terms of glucose concentration, fatty acid concentration, amino acids, so on and so forth. So like insulin, you can see increases glucose uptake, increases glycogenesis, formation of glycogen, inhibits glycogenolysis, inhibits gluconeogenesis. So this table is very good as a summary when you're studying the endocrine system, and specifically the endocrine pancreas, summarizes all the effects of these different hormones. Okay? The last part of this chapter, is there any question on the pancreas or anything we've talked about? The last part of this chapter is on the parathyroid gland and its role in calcium homeostasis. And we've already talked a little bit about calcium homeostasis in this chapter when we were talking about the thyroid. If you remember, the thyroid, in addition to thyroid hormone, releases calcitonin. And calcitonin and parathyroid hormone are antagonistic to each other. They do different things. And they both play a role in calcium homeostasis, but in different ways. And so when I lecture, usually I talk about the thyroid gland and the parathyroid gland at the same time because they play that role, and they're also in the same structure. But this textbook doesn't do that. I'm not sure why. Another reason why I need to write my own textbook, right? But anyway. Parathyroid gland, obviously on the posterior aspect of the thyroid. The thyroid gland wraps around our trachea, doesn't come to doesn't meet in the back, but on that back side there's nodules, usually on the average of 4, which are the parathyroid gland or parathyroid tissue. And what that tissue does is release one hormone and one hormone only, parathyroid hormone. Plays a role in calcium homeostasis. Okay. This says that it regulates calcium to prevent changes in neuromuscular excitability. Well, that's true, but that's a gross oversimplification. It's regulating calcium levels. And as we've seen, and I hope now we can appreciate that we're near the end of our class, that calcium is utilized everywhere, in every cell of our body, oftentimes in the same cell for different reasons. You know, we just saw that the beta cells aren't going to release insulin without it. We saw in the nervous system that neurotransmitters aren't going to release without it. We'll find out in the next chapter, reproduction, that reproduction doesn't happen without it. It's important everywhere. So regulating that concentration, that blood concentration, is vitally important. And calcitonin and parathyroid hormone are the 2 big hormones that do that. Not the only ones, but the 2 big ones. Vitamin D also plays a role. So we have the hormonal effects of calcium availability, meaning the regulation of that blood calcium concentration, maintaining what it's supposed to be, and the actual utilization of the calcium, the metabolism of that calcium in different cells. Okay, and so the regulation of the calcium available, which is the calcium in the extracellular fluid, making it available to those cells that do need it, is a matter of obtaining that calcium from different sources. Obviously the dietary source, but in terms of stored calcium, that would be from bone, specifically compact bone, the kidneys, not necessarily a source, but an avenue to obtain calcium or get rid of it, and the intestines in terms of absorption. Okay. And what parathyroid hormone does at the end of the day is it increases blood calcium. So it is referred to as a hypercalcemic hormone. Okay. And calcitonin, if you recall, is a hypocalcemic hormone. Okay. And a primary source of this calcium released as an effect of parathyroid hormone is at the level of the bone. Our bones are continually remodeling, getting, I don't want to say bigger, definitely bigger when we are children and actually physically growing. But in adulthood, our bones are still remodeling, usually as an effect of making calcium available or storing calcium, but they're continually changing, okay, to release free calcium into our blood or removing it from our blood, okay. And this says parathyroid hormone's immediate effect is to promote transfer of free calcium from bone into the plasma, okay. That's not its only effect. That's like its first one, okay. When parathyroid hormone is released, when we need to increase our blood calcium concentration, its first stop is compact bone, okay? But it's also going to do, it has an effect at the level of the kidneys, it has an effect at the level of the digestive tract, but its first effect is on the bones, okay? So let's talk about this, and then we'll take a break. But compact bone is formed of its functional unit, the osteon. This little thing's in my way. Okay. It's the functional unit of bone up here. Okay, and at the center of these osteons is a central canal. This is where our blood supply is. Okay, and then scattered in this osteon are osteocytes, both osteoblasts and osteoclasts, that are going to play a role in that bone remodeling, okay, bone formation and breaking down of the bone. And so if you look at this illustration, just to get you referenced, here is the osteon itself and the central canal. So we're looking at the center right here, over here. Okay, you can see the osteon is formed, surrounded by these osteoblasts, the bone-forming cells. These are the cells that take free calcium, incorporate those into calcium salts, and deposit that into the compact bone, or further enhance the compact bone by the addition of these calcium salts. And then on the other side, we have the osteoclasts, which actually originate from macrophages that differentiate into these cells. And these are the cells that break down the compact bone to break down those calcium salts and release free calcium into the vascular system. Okay, and specifically what these osteocytes do, or osteoclasts do, is they break down this mineralized portion of bone, the calcium salts, to release free calcium. And there's two ways that the calcium enters into our circulatory system, under what's called fast exchange and slow exchange. And fast exchange is a mechanism where we are initiating a calcium pump. That's why we have ATP down here. Okay, we are pumping calcium against the concentration gradient through a calcium pump that's activated by parathyroid hormone. Okay, and then we have the slow exchange, which is also an effect of parathyroid hormone, but it's the parathyroid hormone action on the osteoclast itself initiating the breakdown of these calcium salts and the passive diffusion of calcium into the circulatory system. But the net effect is to make available free calcium and to bring up that calcium concentration. Okay. So its net effect in terms of making available free calcium at the level of the bone is through the activation of osteoclasts, okay, to increase the calcium levels in the plasma. Now it tells us right down here it acts on osteoblasts, and you might be thinking, well, you just lied to us. You said osteoclasts. Well, it initially activates on osteoblasts to cause them to secrete this compound called rankle. And rankle stands for, I keep wanting to say receptor associated, but it's not receptor. I'm forgetting what the R stands for. But associated NF-kappa B ligand. Okay, so it's going to have an effect on macrophages and the NF-kappa B pathway. But what happens is these osteoblasts and the secretion of this rankle form osteoclasts. Okay, and I put this chart in here, this illustration in here this morning. And what's happening is down here, again, we have our osteoblasts, okay, and the actual compact bone, the bone-forming cells. And what they do is they release this factor called the rankle, okay? And rankle will bind to receptors that get expressed from, you can see this is the osteoclasts originally come from macrophages that differentiate. The rankle binds to its receptor on these cells, and they differentiate into osteoclasts. And at this point, they are activated. And through this association with the compact bone, starts to dissolve free calcium available to the circulation. I got this illustration from, it was actually from a paper in the journal Nature, but it was talking about a new drug, denosumab, that inhibits this stuff. Okay. But in addition to its effect on compact bone, it also has an effect at the level of the kidneys. And what it does at the kidneys is, again, remember that this is a hypercalcemic hormone. It's raising blood glucose. So what it's going to do at the level of the kidneys is to increase the reabsorption of calcium. Okay? And with the reabsorption of calcium, we're also going to be sending phosphate in the opposite direction. So we're going to be reabsorbing calcium, and we're going to be secreting phosphate. Okay? And so it's doing this with an antiport mechanism. But the point is, we are going to be reabsorbing more calcium. So this isn't mobilization of calcium from a storage standpoint, but it's the increased reabsorption at the level of the nephron, where we are taking that free calcium that we would otherwise eliminate in the urine, and now we're going to utilize it in the body to further increase the calcium concentration. Okay. Why don't y'all take a break? Take a 15-minute break, and we'll continue on. We're almost done with this chapter, and then we will take a chunk out of reproduction and call it a weekend. I think PCOM needs a faster entry system. You know, when you use these things, there's like a delay. Every time I'm slamming into the door because it's... Okay. Okay. Is this on? Yeah. Let's continue on. We're almost done with the endocrine system. But like we talked about, parathyroid hormone, it is, its role is to increase blood calcium. It does this at the level of the dissolution of compact bone, the breaking up of the calcium salts, primarily calcium phosphate salts, releasing this free calcium into the circulatory system. It also works at the level of the kidney to initiate the reabsorption of sodium or calcium from the nephron at the same time as the elimination of phosphate into the nephron. Bless you. And it also works at the level of the digestive tract, promoting the absorption of dietary calcium into the circulatory system. So it's this 3-level of calcium regulation, specifically 3 levels of increasing blood calcium. Okay. It also plays a role, or not a role, but it goes hand in hand with the action of vitamin D, active vitamin D3 specifically, which is a hormone, even though it's called vitamin D3, it's calcitriol, that further enhances the process of calcium absorption at the level of the digestive tract. Okay. And if you recall, like we said, when we were talking about the thyroid hormone, we talked about calcitonin. And the relationship between calcitonin and parathyroid hormone is much the same as the relationship between like glucose and glucagon. They do opposite things. Parathyroid hormone is a hypercalcemic hormone, and so calcitonin is a hypocalcemic hormone. And they're the 2 primary hormones that play the role in regulation of blood calcium. And again, there are others. There's more, but these are the primary ones. Okay. Again, vitamin D, specifically vitamin D3, active vitamin D3 or calcitriol, increases the process of absorption of calcium from the gut. Okay. And parathyroid hormone plays a role in the activation of vitamin D. And vitamin D originally begins, the synthesis of vitamin D begins through the absorption of ultraviolet radiation at the level of the skin. Okay. Ultraviolet radiation will transform cholesterol into cholecalciferol, and cholecalciferol gets into our circulatory system, and that cholecalciferol that passes through the liver becomes hydroxylated, and then at the level of the kidney becomes hydroxylated once again, and at that second hydroxylation step, that is active vitamin D3. Okay, and it plays a role in the regulation of calcium at the same time as regulation of phosphate concentration. And this illustration tells you all the things that happen in relation to a decrease in blood plasma phosphate. what it's going to do is signal to the kidneys to activate that vitamin D3, so to hydroxylate that molecule into active vitamin D3, calcitriol. And what calcitriol is going to do, two things. It's going to increase phosphate absorption from the intestine. And if you remember from calcium, that's also going to increase the calcium absorption. And so that's going on as well. So these two, the calcium and phosphate absorption, are going hand in hand as an effect of calcitriol. Okay. And it's also going to, when we have a decrease in plasma phosphate, because calcium and phosphate are being shuttled in opposite directions, you're going to have a decrease in blood calcium, and that's going to activate that system we've just been talking about, the parathyroid system. Parathyroid gland is actually, in this case, with an increase in blood calcium, it's going to decrease parathyroid hormone. And so what that's going to do is, bless you, decrease calcium reabsorption at the same time as increasing phosphate reabsorption. And if we're decreasing calcium reabsorption in the kidney, what that means is we're increasing urinary output. Okay? And in this case, we're decreasing urinary output of phosphate. The net result is to fix the problem, an increase in the plasma phosphate concentration. Okay, so that's going hand in hand with calcium homeostasis as well. And phosphate is obviously important because that is the, the primary component of molecules such as ATP. It's a primary molecule of DNA, so phosphate regulation is very important as well. Disorders of calcium metabolism can be problematic as well. This can also affect the nervous system, but as we've seen, and calcium being so important in our body, having too much or too little can be very disruptive. And we're talking about hyposecretion. And in terms of specific calcium metabolic disorders, we're talking about usually the hyper or hyposecretion of parathyroid hormone. Now, what causes a hypersecretion of parathyroid hormone usually is a tumor on the thyroid gland itself that is impeding the function of the parathyroid gland tissue. Okay. And specifically hyperactivating it and causing it to increase the activity, or not activity, but the production of parathyroid hormone, which will lead to hypercalcemia. And on the other side of the spectrum, parathyroid hyposecretion, hypoparathyroidism, what this is usually caused by is that physical removal of parathyroid gland tissue. Having thyroid cancer, in terms of cancer, is not all that uncommon. And if radiation and chemotherapy don't work, excision of the thyroid gland itself is a last-ditch effort. Whenever possible, they try to salvage the parathyroid gland tissue, if they can, to maintain the tissue that produces parathyroid hormone. But in the case of necessity to remove it, you're removing the source of parathyroid hormone. And that's not to say you can't survive, because that can be given exogenously, as can thyroid hormone and as can calcitonin. Okay, but nonetheless, that is the primary cause of hypoparathyroidism. And obviously, with a vitamin D deficiency, we are going to have a difficult time in absorbing the necessary amount of dietary calcium. Okay, but again, in developed countries, we don't need to worry about that, but they do in underdeveloped countries that don't have organizations like the FDA. All right, that is the end of this chapter. With one chapter to go. Any questions yet? I also wanted to point out that we're in the home stretch, right? We just have the rest of this class and just 2 more. And I know you're probably excited about moving on to the next phase of your PA program. You've all done a very good job. But now that we're moving in the last week, a little reality is going to set in. And you're going to realize, wait a minute, I'm done with human physiology almost. That means I'm done with Dr. Brandon. And then you start to go into, denial a little bit, depression. It's not uncommon. Usually the last week of class, I bring in a box of tissues. Yeah, here we go. And so, if you just feel the need, just let it go. We'll pass them out. We'll do a group hug, whatever. I just want you to know, don't be embarrassed. When those emotions just well up and all of a sudden, so just, I feel you. But the last chapter is the best chapter because it's reproduction. And here it is. We'll be able to make a pretty fair amount of headway into this chapter. We're going to talk about the reproductive system in both males and females, and it will be broken down. We'll have a little introductory stuff first, and then we'll talk about the male, and then we'll talk about the female. From a physiologic standpoint, from a molecular physiologic standpoint, and from a physical standpoint. So, the reproductive system is a system that, believe it or not, we really don't need, unless we want to procreate. Like, we need our cardiovascular system, right? We need a respiratory system. We need all those other systems, but the reproductive system is the only one that we don't need to survive. We need it to procreate, to make more of us, but we don't need it for us as individual units to survive. Okay. I like this bullet list here. It says it plays other roles: psychosocial behavior. I'm not really sure what it's getting to at that. Personal views of self. If you feel bad about yourself, go practice procreating, I guess. But that along the same lines might make you feel bad about yourself. Like, what did I just do? That kind of thing. Interactions with others, societal effects. But I have regulatory mechanisms in bold at the bottom. Because the reproductive system does play a big role in other regulatory mechanisms. Like estrogen plays a big role in bone development. We talked a little bit about bone development in that last chapter, osteoblast activity. Estrogen plays a big role in that process. Okay. Testosterone plays a big role in protein synthesis. And those are just 2 examples. Reproductive function plays a big role in regulatory mechanisms. Okay? So the reproductive system as a whole is composed of, this says the gonads and the reproductive tract. I've combined those 2. The gonads are part of the reproductive tract. And accessory sex glands, which are more significant in the male and much required in the male than the female. But then we're going to go through the male reproductive system and the female reproductive system separately. And we talk about the male system first, just because in this textbook it's the first, but I always talk about the males first, not because, like we're number one kind of thing. Honestly, the male reproductive system is a lot less interesting than the female reproductive system for all kinds of reasons. And so I always talk about the guys first. This illustration of the male system, this is always a little uncomfortable for me as a guy because it's just taking a slice right through the package there. It's just a little uneasy. And yes, this is called the package. That's an anatomical term that I invented. Why isn't my thing working now? That's not a good way to phrase it when I'm talking about the male unit there. It's called the package, and it's separated into what's called the unit and the satchel. Those are my own anatomical terms. But nonetheless, this is the male reproductive tract beginning down at the level of the gonads, the testicles, the structure behind the testicles called the epididymis, the vas deferens, which is the avenue that sperm begin to make the process of emission from the body, up through the vas deferens around the urinary bladder. And of course you've got vas deferens coming from both testicles, so this is just, you're seeing one side of it. And then they come together, they merge, the two vas deferens merge together in what's called the ejaculatory duct. And on either side of the ejaculatory duct are two of the first accessory sex glands, the seminal gland or seminal vesicles. And then the sperm move through what's called the prostatic urethra, and the prostate itself is also an accessory sex gland. And then it continues on into the penile urethra, and you can barely see it, but two small glands on either side of the prostatic urethra, right at the junction of the prostatic urethra and the penile urethra, are the bulbourethral glands that are the last set of accessory sex glands in the male. And then it's out the body. Okay. And then on the female side, this might make you ladies uncomfortable, I don't know. But again, starting with the gonads, obviously in the female, the gonads are internalized structures, and they are right above the uterine body itself. And it's associated with the oviduct, the fallopian tubes. There's not a direct physical connection. There's actual physical space in between the ovary and the oviduct. And the oviduct, the tips of the oviduct, the top, the most proximal portion of what's called the infundibulum, which is the first section of oviduct, kind of wraps around the ovary. Okay, and if you looked, if you took a if you went into the abdomen, the lower pelvis actually of a female, looking at the reproductive tract, you wouldn't even see the ovaries. They'd be within the fimbria, those finger-like projections that wrap around the ovary. And when ovulation occurs, there's really only one place that egg can go, even though for a period of time it's in the, I guess you could call it in the abdominal space. Okay, but then it continues on through the oviduct, fallopian tube, down to the uterus proper. The uterus is a very strong muscle. It is used for one thing and one thing only, and that is gestation, developing of the early embryo / fetus, and that's its role. And it's a big muscle because it's the muscular contraction used for expulsion of the fetus. and it's made up of smooth muscle, and it's made up of 3 layers of smooth muscle, and those 3 layers go in different, it's oriented that they go in different directions. So when that muscle contracts, it really contracts. Okay. And then we move into the vagina and out the body. And you can see the uterus overlies the urinary bladder. And so this is a good illustration to show you why when women are pregnant, they have to go to the bathroom frequently, because as you have that developing fetus, is pushing down on this urinary bladder. This is another good illustration showing how these structures are associated with each other. And whenever you see illustrations like this, I always remind students that it is the reproductive tract has been kind of stretched out so you can see all the components of it. In real time, it's all in there together. And again, this is a good illustration of the first section of oviduct, which is called the infundibulum. And this is where fertilization usually takes place, way up in here. Okay. The cells of the reproductive system are the sperm and the egg, okay, the gametes. And the gametes are the only haploid cells that we have in our body, the only cells that have half the chromosome number. In the human, we have 46 chromosomes, or 23 pair, however you want to refer to it as. In the somatic cells of our body, but in terms of gametes, we have haploid cells, so that when we have fertilization, we have a haploid sperm with 23 chromosomes uniting with a haploid egg with 23 chromosomes, so we have the full complement of 46. Okay? And these are the only cells that go through the process of meiosis, not mitosis. Well, there is mitotic division, but the production of these gametes is through meiosis, reduction division, where we go from a diploid cell, a cell that has the full complement of chromosome number, and we reduce that number in half. That's why it's also called reduction division. And the process of production of these gametes is collectively gametogenesis. Okay. And also, as an effect of meiosis, is we have genetic differentiation. Genetic variation goes on through meiosis, early on in meiosis. And so that the resulting gametes are genetically different from the parent cell. You know, mitosis gives us cell division, but it gives us cell division and resulting 2 cells that are genetically identical. They're carbon copies of each other. Every meiotic cell division, we are having genetically different cells. So every sperm cell in my body is genetically different from each other and genetically different from my somatic cells because of the genetic variability that occurs in the process of meiosis. Okay. And in terms of sex, when you have fertilization, what determines the sex is actually the sperm cell. Because the eggs, the oocytes, they contain, in terms of sex chromosome, okay, in the chromosome array in the human, we have what are called 22 autosomes, 22 pairs of autosomes, and a pair of sex chromosomes. Okay. And in the egg, after meiotic cell division, the egg possesses an X chromosome each and every time. And the sperm are going to either have an X chromosome or a Y chromosome. And so if an X-bearing sperm fertilizes an egg, it's going to be a female, XX. And if a Y-bearing sperm fertilizes an egg, it's going to be a male. It's going to be XY. Okay? And so it's the presence of the X or Y chromosome in the sperm, in terms of sex chromosomes, that determines the sex of the individual. Okay, and the process of sexual differentiation occurs at that point. Well, begins to occur. Okay, the differentiation of the at that time sexually undifferentiated early, I guess, fetus. We're past the embryonic stage. It's neither male nor female. But when the genes, that Y chromosome or the X chromosome starts to become active, then the reproductive structures start to develop. And this is early on in embryonic development. Okay? And again, it's all based on the presence or absence of that Y chromosome. Okay? And what that initiates is the differentiation of the gonadal structures, and we'll talk about that in a second. Okay? And then the development of the interior and exterior genitalia. Okay, now this is from your textbook, and this shows you what happens in terms of sexual differentiation, but I put in a couple more from my own lectures that are just easier to see. This is a very informative slide, but it's real busy, and so I'm going to proceed into some slides I put in from my lectures. But what determines the actual sex of the individual, again, is the presence or absence of the Y chromosome in the sperm cells themselves, and more specifically, the genes in that Y chromosome. Okay? We're going to see a picture of side by side the X chromosome to the Y chromosome, and the Y chromosome is very small, and there's very few genes on it. But there's a few very important genes, and probably the most important gene in terms of sexual differentiation is a gene called the SRY gene, and that stands for sex determining region of Y. Okay, and what this gene does is it codes for this protein called testes determining factor. I don't know why this thing doesn't write sometimes, and sometimes it does. But nonetheless, testes determining factor, and its name kind of tells you what it's going to do. It's going to start development of the male reproductive tissues, specifically the testes. And early on in embryological development, you can see 8 to 9 weeks, we start to secrete testosterone. Now, prior to this point, the reproductive tissues are going to originate from a structure called the gonadal ridge. And I've got a picture to show you all this in here in a second. Okay? And in that gonadal ridge, you have tissues that are named the Wolffian duct and the Mullerian duct. And if the SRY gene is present, and thus it becomes active, and starts to produce this testes determining factor, that stimulates the Wolffian duct to develop. And the Wolffian duct is going to develop into male reproductive tissues. And at the same time, it inhibits the Mullerian duct, development of the Mullerian duct. Specifically, there's a factor produced called Mullerian inhibiting factor that does as its name implies. Okay? And on the opposite end of the spectrum, if the Y chromosome is not present, what don't you have? You don't have the SRY gene. So you don't have that testes determining factor. And you don't have that inhibition of the Mullerian duct. So in that case, the Mullerian duct develops over the Wolffian duct. And female reproductive tissues will develop. So it's it's the Y chromosome with the SRY gene that determines the sex. Okay. This is what the human chromosomal makeup looks like. This is called a karyotype, and it's a good way to look at all the chromosomes at one time. And what you do is you take a cell and you stimulate it to go into the S phase of the cell cycle so that we have the synthesis of DNA, and we have the sister chromatids, and then there's stains. You can look at different regions. And just by eyeballing a karyotype, you can tell, number one, what organism you're looking at, and we know this is a human because it's got 23 pairs of chromosomes. And we also know it's a male because down here, the sex chromosomes, we have an X and a Y. And it's very easy to differentiate the two because the Y chromosome is tiny compared to the X. And I also have this picture. This is a scanning electron micrograph of the X chromosome and Y chromosome. And I always point this out to students. And I tell them to look at that X chromosome. And obviously it's a sister chromatid, so it's the chromosome that's just copied itself. But look how pretty that X chromosome is. It's long and slender and large. And then look at the Y chromosome. It looks like a little booger, doesn't it? It's kind of a blob sitting there. That's the guy. That little thing. And again, there are not many genes in there, but there's the SRY gene. So even though it's a little booger, a little blob, it's kind of important. Anywho, with the production of the SRY gene product, the sex-determining region of Y chromosome and the SRY gene, we have the production of the testis-determining protein, okay? And that's going to stimulate the gonadal ridge, more specifically stimulate the Wolffian duct to begin to develop, and specific cells get stimulated. We start the stimulation of the Leydig cells, and if you remember, these are the cells in the testicle that produce testosterone. And this testosterone is going to begin the production, or the development, I should say, of the Wolffian duct. And eventually we're going to develop into, that's going to develop into male reproductive tissues. And at the same time, another cell type called Sertoli cells, and even though we're talking about an early embryo, these cells are going to be very important through the life of the individual. These cells are going to be the source of testosterone for the life of the individual. Not the sole source, but the primary source. And Sertoli cells are also going to play an important role, and we'll talk about those later. But in this case, what the Sertoli cells do is they secrete that inhibiting factor. That factor, or Mullerian inhibiting factor, that causes the inhibition of that Mullerian duct from developing. So male tissues develop, okay? And then again, what causes the female tissues to develop is the absence of that Y chromosome. So an X chromosome bearing sperm. So you don't have that SRY gene, you don't have that testis determining factor, you don't have that Mullerian inhibiting factor, and the Mullerian duct develops. I used to always make fun of my friend at the vet school, whose name is Eric Mueller, and you know, the Mullerian duct, so I told him he was named after female parts. But now he's my friend who lost his wife last week. That's who got killed in the car wreck. So I'm no longer going to make fun of him. From an embryological standpoint, these are the tissues that begin developing at early embryonic development, just shy of 2 months. We just have tissues that are going to that can become either or, and they're going to become either or based on the presence of that SRY gene or the absence of that SRY gene. Okay. In the presence of the SRY gene, early on, just a couple weeks later, we have the beginning of the development of the structures into the male parts. And at this point, we just have just a little knob that's going to be the beginning of the penis, and the beginnings of the urethra. and scrotum. Okay, and then just around the time of birth, we have the descension of the testicles into the scrotal area. They begin development intra-abdominal. Okay, and the penis. Okay, and the foreskin, and then again, in the absence of the SRY gene, we have the development into the female tissues. But it's all based on the presence or absence of the SRY gene. And this is just another illustration showing that from another angle, if you will. The Wolffian duct and the Mullerian duct are here together in the undifferentiated embryo. It's not showing another structure that is called the gonadal ridge that adjoins these two structures. And again, in the case of the male, the Wolffian duct develops. In the case of the female, the Mullerian duct develops. And this is just another representation of that. And I just put this in because this actually shows the gonadal ridge here on either side. Okay, so male reproductive physiology. We talked about where the male gonads come from. Well, both of them come from the gonadal ridge. And again, the beginning of the testicles, throughout the period of gestation, throughout the period of time where we are inside our mom, for males, the testes are still intra-abdominal, and they don't make that descension into the scrotum until right around the time of birth. Okay, just before or during or maybe just after birth, but right around that time. And it's very important that they do make that descension into the scrotal area and remain there, because we'll find out that spermatogenesis, well, here it is right at the bottom, spermatogenesis occurs at a lower temperature than core body temperature. It's about 1 degree centigrade, which is a very subtle difference, but it's a critical difference. Spermatogenesis will not occur in testicles at core body temperature, and so they must be extra-abdominal. Okay, and failure of a testicle or both testicles to descend is called cryptorchidism. And even though spermatogenesis doesn't occur until puberty, so you know, in the human 11, 12, 13, maybe older, spermatogenesis doesn't occur then. If the testicles remain in the abdomen for any period of time after birth, the individual will be sterile, or that testicle will be anyway. It's a big problem. Well, not a big problem, but in horses, if a stallion has, what just happened? Is that because I lifted that thing up? If a stallion has reproductive value, breeding value, and their cryptorchid is a colt, you have to correct that right away. And it's relatively easy to correct, but it needs to be taken care of. But anyway, that's why the male gonads are extra-abdominal and remain in the satchel, in the scrotum. If you take a cross-section of the testicle, what you will see from side to side, top to bottom, are circularized structures called seminiferous tubules. And I haven't seen a picture of a micrograph of that in this chapter, and if there aren't any, I'll put some in just so you can see what they look like. But nonetheless, they're circularized structures and called seminiferous tubules, and that's where spermatogenesis occurs. And between the seminiferous tubules are cells called the Leydig cells that are the source of testosterone. They're also called interstitial cells because they're in the interstitium in between the seminiferous tubules, but that's the source of testosterone. And testosterone along with FSH from the anterior pituitary are the two hormones required for spermatogenesis to occur. Okay. And also testosterone, in addition to its role in spermatogenesis, is very important for, like we said a minute ago, protein synthesis, for development of secondary sexual characteristics, you know, facial hair, pubic hair, axillary hair, you know, increase in muscle mass due to its, and obviously we're talking about puberty, pubertal changes, increase in muscle mass because of its significant effect on protein synthesis, you know, deepening of the voice, all that stuff. Non-reproductive actions can be, an example can be its effect on protein synthesis. Okay, and these reproductive hormones come from the same parent molecule. I think we talked about it before, but it all comes from cholesterol. And the metabolism of cholesterol is where we get our sex steroid hormones. And so testosterone, even though we regard testosterone as a male hormone and estrogen as a female hormone, we all have the same hormones. The males have more testosterone than the females, and vice versa. And these hormones can be converted from one to the other through different metabolic pathways. If you take testosterone and put it in the presence of an enzyme called aromatase, it's going to become estrogen. Okay? So there is an interconversion. So effects of testosterone before birth, we talked about that. It causes the development of the Wolffian duct into the male tissues. Okay? In puberty, it causes growth of the reproductive system and the individual as a whole. It's absolutely required for spermatogenesis, and we'll talk about specifically where or at what level it's required. Maintains reproductive effect, development of different secondary sexual characteristics, and then again, non-reproductive functions, protein synthesis, bone growth at puberty, well, a lot of growth at puberty. And that includes closing, remember we talked about the epiphysis and long bones. It closes that epiphyseal plate. So it plays multiple effects at different times over the life of the individual. Life meaning even prenatal, okay, prior to birth. Spermatogenesis is the process of obtaining the gamete, and we're talking about meiosis, meiotic cell division, reduction division, but it actually begins with mitotic cell division. Let me just draw for you really quickly. Where are the markers? Oh, there you go. If you look at the cross-section of a testicle, you'll see a bunch of these, and they're called seminiferous tubules, okay, all across the testicular tissue, okay. And on the periphery of the seminiferous tubules, we have the germ cells that are going to give rise to sperm cells called spermatogonium, okay, and they're diploid. They have the full set of chromosomes. And what happens initially is when this is stimulated to begin the process of spermatogenesis, and what initiates it is FSH, follicle-stimulating hormone. So at puberty, these spermatogonium will begin the process, and the first thing they do is they divide mitotically. And one spermatogonium is going to become two. Okay? And one spermatogonium stays behind, and it's called maintaining the spermatogonial pool. So we never run out of sperm cells because we never run out of our progenitor cell. And then that other one is going to start the process of meiotic cell division and migrate through the tissue of the seminiferous tubule. And all the way until the very end of meiosis even, it still looks like a cell, a round cell. It doesn't look like a sperm cell. It's not until the very end that it differentiates into what looks like a sperm, and it's still attached to the seminiferous tubule itself, but then detaches into the lumen and then is washed down to its next location. Okay? So the first division is actual mitotic division. But then the process of meiosis begins. And I'm not going to go through the specifics of meiosis because I think we've all been through that. But I just want to point out some of the inherent differences between meiosis and mitosis, like reduction division, like genetic recombination. Okay? So this occurs in the seminiferous tubules and also requires the cells in between the seminiferous tubules, called Leydig cells, that produce the testosterone, and cells within kind of the seminiferous tubules called the Sertoli cells. Remember, we saw those at the beginning of sexual differentiation. The Sertoli cells are the ones that secreted that Mullerian inhibiting factor. Well, now they're going to play a big role in spermatogenesis. They're also called nurse cells because they kind of take care of the developing sperm cells. Okay. And so this, again, is a process of spermatogenesis. Beginning at the, again, periphery of the seminiferous tubules, we have the diploid spermatogonium, initially dividing mitotically to give two daughter cells, genetically identical, because this is mitosis, and one of them will remain behind to maintain the spermatogonial pool, and the other one's going to begin the process of spermatogenesis. And you can see here the next division, again, is mitosis. But it's from here that we begin the process of meiosis. And again, another inherent difference between mitosis and meiosis. Mitosis, we divide once and we go from a progenitor cell to two daughter cells, genetically identical. And we're done. Meiosis, we're going to have two cell divisions. So in the process of meiosis, we're going to go through one, you can see reduction division. So here we have the chromosome number, and we end up with two cells. And then these two cells will divide again to give us four cells. So in the process of meiosis and spermatogenesis, we're going to go from one progenitor cell to four different sperm cells and four genetically different sperm cells. And you can see down here we have these cellular structures called spermatids, and we're done with meiosis. And we have the four resulting sperm cells, but they don't look anything like a sperm. Okay? They are residing at right at the lumen, the junction of the lumen in the seminiferous tubule. And then at that point, they go through another process called spermiogenesis, and this is where they undergo the morphological change, going from that round cell structure of the spermatid into the spermatocyte. And then we'll see where they go from here. But I want to point out at this time, even though they look like a sperm cell, they have the head, the tail, and a structure called the midpiece. They're anatomically correct, but they can't do anything. Bless you. They can't swim. They can't fertilize an egg. They can't do anything. Okay. Hormones required for this process are the same hormones that are required for the reproductive activity of the female, again from the anterior pituitary and hypothalamus. GnRH, which regulates the production of FSH, follicle-stimulating hormone, and luteinizing hormone. Again, the follicle-stimulating hormone is required for spermatogenesis, and we'll see specifically where in a second. And the LH, luteinizing hormone, is required for testosterone production from Leydig cells, which is also required for spermatogenesis. Without those two hormones, we don't have sperm cells. Okay? There is feedback control, a negative feedback mechanism to regulate testosterone levels, but it's a very weak negative feedback role. In other words, when the human male reaches puberty, production of testosterone begins with Leydig cells, and it continues through the life of the individual. Testosterone levels gradually decline after around age 21, 22, but they're still there, and it still plays a role in reproductive function. Okay? There is a feedback effect of testosterone and inhibin that we also have in the female that inhibits FSH and LH and all that stuff, but it's a very weak regulatory role. It's very profound in the female because she is going to do two different things on either side of ovulation. On the one side of ovulation, she's developing an egg, really. And then on the opposite side of ovulation, she's preparing for pregnancy. And that's all under hormonal control of vastly different hormones doing different things. So it's a very dynamic situation in the female, and the feedback regulation of FSH and LH is vitally important and a significant feedback effect where in the first part of the cycle, prior to ovulation, FSH, follicle-stimulating hormone, predominates, and right before ovulation, LH spikes up. And then in the latter half of the cycle, those two hormones are baseline, and they are inhibited absolutely by, some other hormones. I don't want to jump into the the female reproductive section right now, but there's a very powerful negative feedback effect in the female, not so much in the male. Okay. Specifically, where the testosterone and FSH play a role is the testosterone plays a role in the initiation of the spermatogonium, the germ cell, undergoing mitotic cell division and initiating the process of meiosis. And the big role of FSH is in that process of spermiogenesis, the transformation of that spermatid, that haploid spermatid that looks just like a cell, its transformation into a anatomically correct sperm cell. Okay? And this is all established through the link up of the hypothalamo-pituitary gonadal axis. Once you have communication between the hypothalamus and anterior pituitary and gonads in both sexes, that's puberty. Okay, you've hit puberty. Now you have the hypothalamus talking to gonadotrope cells, the anterior pituitary, to produce gonadotropins, gonadotropic hormones. And those gonadotropic hormones start to do its thing on the gonads. And in the male, it's the FSH stimulating spermatogenesis, and it's the LH stimulating testosterone production. And once you have that axis wired up, you've hit puberty, and you know, you're reproductively active for the duration of your life. Okay. And like I said, testosterone and inhibin, and we're going to see this hormone in the female as well. These play an inhibitory role on GnRH and FSH and LH secretion from both sides, from the testosterone and from the inhibin. I always think this is kind of ironic. The process of GnRH production begins with the activation of the KISS1 neuron to produce something called kisspeptin that starts GnRH. Starts that reproductive function. It's called KISS. Yeah? No? Okay. Never mind. Again, at puberty, we have something in the hypothalamus called the GnRH pulse generator, and it generates pulses of GnRH that stimulate the anterior pituitary to release their gonadotropins. Okay. Melatonin, remember we talked about that in the endocrine chapter coming from the pineal gland. I said it really doesn't play a big role in the human, and it really doesn't in the adult. It does play a big role in the timing of puberty. And again, it plays a big role in lower animals that are responsive to light, more so than we are. Melatonin specifically is an inhibitor of GnRH. So melatonin has the ability to inhibit the whole reproductive system. If it can inhibit GnRH, it's going to inhibit FSH and LH, which is going to inhibit what it does at the level of the gonad. But levels decline at puberty, and so that inhibitory role is relaxed, and then you start to get GnRH. And then you start to get FSH and LH, and then you get the development, or the initiation of spermatogenesis, in the male anyway. Okay? And what happens in the process of spermatogenesis is the sperm concentration is increased significantly over time. And where this structure down here called the epididymis is where these sperm are going to wash down from. When they're released into the tubule of the seminiferous tubule, they'll eventually wind up in the epididymis, which is a structure on the posterior aspect of the testicle. Okay, and from there they're going to go into this is called the ductus deferens. It's also referred to as the vas deferens, and that's what I call them. And it's that structure that's specifically ligated, cut in the procedure of vasectomy. The accessory sex glands in the male are extremely important for the fertilizing capacity of sperm. Okay? Without the contributions of those three accessory sex glands, the two seminal vesicles, the prostate, and the two bulbourethral glands, without the contributions of the fluids that come from those glands, sperm will not be able to fertilize an egg. Even though they look like a sperm, you could take one out of a testicle and throw it on an egg and it wouldn't know what to do. It can't even swim at that point. Okay? And the process of that biochemical change, where the sperm obtain fertilizing capacity, is called capacitation. They've obtained the capacity to fertilize, which just happens to be an area of research I'm in. And when you see this word semen, you know, a lot of times semen and sperm are used interchangeably, but they're really two different terms. Sperm refer to the cell. Okay. Semen is the sperm and the associated fluids that come from the accessory sex organs. Okay. And those fluids are vitally important to the fertilizing capacity of the sperm. Okay. Now, this table tells you what all these structures, what they produce, where they're located, but I want to talk about the accessory sex glands, and so we're just going to talk about them individually. But they are, again, the seminal vesicle, the prostate, and the bulbourethral gland. I told you a minute ago that testicular sperm, they look right, but they can't do anything. They can't swim. They can't swim until the process of ejaculation occurs and the sperm cells get far enough into the tract where they have just bypassed the seminal vesicles. Let me just go back to, got to go kind of far back. Wait. Yeah, this one. Let me see if I can draw now. Yeah. So we have the two vas deferens, ductus deferens, and you can see they come to a point where they merge into the prostatic urethra. And at this point, we've just gone by the two seminal vesicles. And the seminal vesicles contribute the majority of the ejaculatory fluid, about 60%. And there's many different components of that seminal vesicular fluid that's critical for the sperm. And the first one is it's rich in fructose. And fructose is a carbohydrate that sperm use for cell respiration. So it's at this point where they get their potential energy source that they can use to obtain ATP so it can swim. So it's not until this point that they are able to start swimming, although they don't really start swimming yet. They're being propelled through a pressure increase. But it's at this point we get seminal vesicular fluid. In addition to the fructose, it's also got, it's rich in fibrinogen. And if you remember what fibrinogen did in the blood, it causes hemostasis. It turns into, when it becomes activated, it turns into that insoluble protein fibrin and causes coagulation. It does the same thing in the sperm. Initially, at ejaculation, the sperm will coagulate. And that's of benefit to them because it kind of protects the sperm and it helps them travel in mass, even up through the vagina if you're talking about specific reproduction with fertility as the goal in mind. The sperm will travel in these clumps. And then there's an enzyme later that will break up that fibrin, and then they start moving individually. Another important component of the seminal vesicular fluid is a protein called seminal plasmin, and it's a naturally occurring antibiotic. So the ejaculate has antibodies in them. Okay, and then it moves through the prostate, and the benefits of the prostate are. Let me go back to that slide we were just at. No, not that one. Yeah. We're almost out of time. The important components of the prostate is, number one, it secretes an alkaline fluid. And that's important too because, you know, now we're entering into the urethra, right? We haven't hit the penile urethra yet, but what usually goes through the penile urethra? Urine. And urine's acidic. And that acidic environment will kill on contact sperm cells. So it's very important to buffer that environment. And there's an alkaline fluid from the prostate that does buffer primarily initially the prostatic urethra, but also the penile urethra too. Okay, and also it provides a buffering capacity for the vagina as well because that is also acidic. And another reason why the agglutination of sperm cells is so important because when they're in these clumps, they're protected to a point from that acidic environment. Also, the fibrinogen is activated, and the fibrinogen comes from the prostatic fluid. Okay. And then lastly, the two small in the human anyway, bulbourethral glands, is that it secretes mucus as well, and also it's very alkaline, and this buffers the penile urethra in addition to the prostatic fluid. Okay, you might have heard of that term pre-ejaculate. The pre-ejaculate comes from the bulbourethral gland. Okay. And those are the importance of the accessory sex glands. Without the seminal vesicular fluid, sperm aren't going to be able to swim. They don't have that potential energy, the fructose. They don't have the antibiotics aren't necessary for the survival, but the fibrinogen's not going to be added. The buffering capacity from the prostate, the buffering capacity from the bulbourethral. There's an enzyme from the prostate that becomes active later that degrades that fibrin and breaks up the coagulated mass of sperm cells. Okay. And so I'll stop there, but would you like me to tell you one more story that has to do with something that we're just talking about, specifically with the bulbourethral gland? Not many people know. Oh, I should go off mic.